This podcast was made possible by CIUT FM.
For decades, we believed gene regulation in yeast followed a simple rule. Transcription factors act like switches, binding near genes to turn them on or off. Activators activate, repressors repress, all right next to their target genes. But a new study challenges that model. Researchers mapped where nearly every transcription factor in yeast binds and compared that to which genes they actually regulate, and the results were shocking. Most transcription factors aren't specialists, they can both activate and repress. Even more surprising, many regulate genes they don't even bind near. Instead, they act from a distance through still unknown mechanisms. The paper we're discussing today rewrites the textbook on gene control, revealing a system far more complex than we imagined. Join me in my conversation with the researchers behind this paper. Well, guys, welcome to the show.
It's great to be here. Thanks for inviting us.
Thank you so much for coming. Can you give us an elevator pitch of the paper without going too deep into the details?
Well, I think one conclusion is that the way transcription factors regulate gene expression is a lot more complicated than we thought. And even in a simple eukaryote like yeast, it's very complicated how transcriptions end up finding their way to the DNA. And then once they're there, how the transcription factors cooperate with each other in order to give regulation. So when we started, we didn't imagine that it would be this complicated.
Yeah. So maybe Lakshmi, can you give us a before and after this paper? What was the general perception before publishing this paper about how transcription factors work, and how did it change after this paper?
Yeah. Well, before I started this work, and of course, many people in the field have this traditional model where transcription factors means they recognize sequence-specific DNA using something called motifs, using their DVDs. And when they bind, they are expected to regulate the downstream gene. And there is a good overlap between the binding and regulation. This is what at least I read in the textbooks and I thought of from the literature. But after I started this project and start gathering the evidence and integrating both the binding and regulation, now I know that this is not a complete story. Like Steve just said, it's much more complex than it is. And binding is not always can be predicted by motif. And
So is this specific to yeast, or if we thought about this model as a general for all the eukaryotes?
I know that in yeast, this is most dominant model that we have read. In higher eukaryotes, it is still the case, but there are some other evidences where binding is not always correlated well with the regulation, but not in a scale that we observed it in yeast. Of course, it's known for few examples. Yeah.
Yeah, there have been papers that have said binding and gene regulation for transcription factors, they don't always overlap, but there were some limitations in the way those papers address the issue. And, for people who've grown up, transcription factors bind to their motif and then they regulate the nearby gene always. You have to have really good evidence if you're going to try and convince people that there's something else going on in addition to that. So we did find many examples of the traditional model, factor binds, regulates the nearby gene, but we found many more examples that didn't work that way.
And how did you guys decide to go with yeast as your model?
Oh, I know this because this lab has been working on yeast before too. And this is an extension of that work. But the reason to why yeast, previously in the Steve's lab, they were working on how coactivators work at different gene classes. And they suggested that these cofactors work at these set of genes and the other way around. But following up with that project, they're interested to see how transcription factors, there might be two sets of transcription factors too, working at different promoter sets. So that's one of the question that we wanted to address when we first started this project. And the other one is that then Steve and his lab was working on various activators and wanted to see how they regulate genes, but they kept looking at only one set of activators called acidic activation domains. And they also
Huh.
want to see if what's the other set of the transcriptional activators and how it acts in the gene regulation.
Yeah, and so yeast, for example, has advantages like there's only 150 roughly transcription factors, whereas humans have 10 times more than that. So having a limited number of factors in a system that in many, many respects, it's very conserved with, say, how humans transcription works, yeast was kind of an ideal system for us to like look at the whole set of transcription factors and how they work together.
Right. So I think you kind of touched on this one, but can you tell us about the origin story of the paper and like how did you guys decide to do this like very comprehensive project in the first place, and how does it fit into the overall research direction of your lab?
Well, so the reason we wanted to do this was, I think, as Lakshmi said, we had found that there was basically two kinds of genes. I'll just simplify and say highly regulated genes and constitutive genes. And we thought that there must be two different kinds of transcription factors, that preferentially worked at one or the other. But how do you find those? When we or other people in the field were looking at factors, they were always the kind that seemed to work at the highly regulated genes. So rather than pull out each one of the 150 guys one by one and look at it, which takes forever, it seemed like a much more efficient process to just look at all the genes at once and try and figure out which ones preferentially work at one class of gene versus the other. And then the other reason, Lakshmi came to the lab and she's incredibly fearless. So somebody who knew more at the start probably would have said, that's ridiculous. I'll never do that. But she was like, yeah, I want to do that. And then after she got all the binding data for where each factor binds to DNA, said, it would be a lot more powerful if we knew which gene each thing worked. And she's like, yeah, I want to do that too. So
Oh, interesting. So the initial project was kind of like just what you did for figure one, just binding.
Yeah, that's how it started.
That's interesting.
Yeah. Well,
Um,
what motivated me was like earlier models with how TFs works was based on strong activators like GAL4 and GCN4. But I wanted to see if this model holds true with the whole diversity of transcription factors too. And that's one of the things that I wanted to see. And that can only be achieved if we get a full picture from all the transcription factors. Yeah.
Yeah, so in the end, we did find factors that worked preferentially at one class of gene than the other, but then all these other things opened up that seemed even more interesting. So that's why there's not just figure one, there's figure 2, 3, 4.
yeah. Yeah. I'd love to just like dive into the experiments with you, but I think for us to really understand what you guys did, let's just take some time and talk about the methodology behind your experiments. I specifically picked three kind of like technology that you guys used: check-seq, auxin-induced Degron system, and 4TU pulse labeling. So let's start with check-seq. Can you give us a brief overview of check-seq, and also why did you not just use chip-seq?
Yeah, sure. So check-seq, the way it works is if we want to map binding sites of a particular TF, the TF of interest is fused to monococcal nucleus. It cleaves the DNA, right? But under regular conditions, it's fine. But when we add calcium, the MNase gets activated and it cleaves the DNA wherever the TF goes. So when we sequence those DNA fragments, we get to know the precise location of that particular transcription factor. So this is the way CheckSeq works. This is first developed in Lamley Lab and later adopted by Henikoff Lab for genome-wide studies. So,
which is coincidentally is across the hall from us. So that
this is quite a simple and easy technique, but at the same time very strong and sensitive. Like the TF doesn't necessarily need to directly bind to DNA for us to able to map its binding. Even if it's not directly binding, we can still assay it by CheckSeq, unlike ChIP-seq. Like you asked why we specifically chose this, and that's one of the reasons. And traditional sequencing methods like ChIP-seq have a low signal to noise ratio, and it might also have some non-specific cross-linking issues. So, and that's why we chose CheckSeq, because it seems like more sensitive and might be able to capture both stable and transient interactions.
Yeah, I think one, just to full disclosure, there are some drawbacks potentially to check, and that is to get the MNase cleavage, the DNA has to be accessible next to where the transcription factor is binding. So if there's like a nucleosome right there, it's not going to cut. So it works best in promoter or gene regulatory regions where there's usually a nucleosome depleted region. So that's one thing. And then the other thing, it's really sensitive, but is it too sensitive? So like if you think about how does a transcription factor find its site on the DNA, it's probably bouncing around sampling different sites. And are we only measuring this very stable things? Or are we measuring transient things or things in between? So we have to keep in mind, what kind of binding we're looking at. CheckRick can't really distinguish that, but I think the methods we're using, we're looking at signals 10 times higher than the background. So hopefully we're getting reasonably strong signals there. And Lakshmi also did CHIP too, so it's not like we just ignored
Yeah.
CHIP, right?
I want to talk about that as well. So you have this like figure in, I think it's one of the supplementary figures where you compare ChIP-seq and CheckSeq, and there are some domains that only ChIP-seq pick up. And so how does that happen?
Okay, I think you're talking about why we are seeing ChIP-specific peaks and
Yes.
CheckSeq-specific peaks.
I think you had this like Venn diagram where like some binding sites were only picked up in the ChIP-seq experiments and not by the CheckSeq.
Yeah, like I said, each technique has its advantages and disadvantages. CheckSeq, like I said, it's more sensitive and it might be capturing dynamic interactions too. Whereas ChIP-seq, it's capturing more stable interactions, and when there is if there is no access to that DNA, MNase might have failed to cut it. That's why we couldn't find it in Check. Whereas in ChIP, it has nothing to do with accessibility. If it's directly binding to the DNA, it can still cross-link and it can detect those sites. So this could be one of the reasons why we are seeing exclusively ChIP sites or Check sites. But other than that, we might be finding some non-specific sites in either of the techniques.
But I should also mention in the paper we talk about, there's a large data set from the Pew Lab where they did ChIP on many transcription factors. And when we use their data instead of the Check data, I mean, all the conclusions are basically the same. So our results aren't dependent on the fact that we use that technique.
Right, because I was going to ask, like, did you get any like pushback from the reviewers that maybe CheckSeq is too sensitive and this is why you're seeing this like random binding pattern?
Well, even before reviewers pointed that, we questioned that ourselves, and that's why we always compared it with other large data sets coming from ChIP-X. So
Yes, we've had pushback in previous papers, but not this one. I think it's pretty well accepted. There's there's a newer technique called cut and run. I don't know if you've heard of
Okay.
that, but it's basically a more advanced thing that works really well in higher eukaryotes. And so I think with that technique, a lot of people use it now. And so the idea that you have a transcription factor one way or the other fused to something that can cut the DNA, I think it's fairly well accepted that that's a
Uh-huh.
totally reasonable technique. Yeah. But in the
Yeah, let's
early days, people were asking the same
yeah.
kind of question you just did.
Mhm. Okay, so I'm just wondering how new is this check technology? Like, because I mean, I have no idea. Is it like fairly new? Like, is it like I don't
Oh,
even. Do you guys know?
you know what a Lemley gel is? Uh, uh, it's like,
No.
SDS gels where you have like
So my background is in math, so I kind of
oh,
have no
okay.
idea
I
what
can't
you guys are
remember the
talking.
year. It's at least 20 years ago. There's this guy, Lemley, in Switzerland, who's
Uh-huh.
like done a lot of technological innovations, like modern kind of
Uh-huh.
protein gels. And so they came up with this method for fusing a transcription factor to eminase. Only they just were doing like one factor at a time and not doing
Mm-hmm.
like genome-wide sequencing. And so then maybe 10 years ago, the lab across the hall, the Hanikoff lab, extended this technique with, genome-wide seq, right? To map factors binding everywhere in the genome, not just at one location where you could check it by PCR. So it's the version that we're using now, it's been around for at least 10 years.
Right. Let's move on to the auxin-induced Degron system. What is it, and how did you guys use it in your experimental design?
Oh, so I would say that, ever since I started the lab, we've always been looking for a way where you could rapidly inactivate a transcription factor and
Mm-hmm.
find out what are the consequences of that. And that goes from like temperature sensitive mutations or deletions of the gene if it's not essential to, adding a drug to try and inhibit things. And all of those are really hard and they have various drawbacks. So this technique was invented about 10 years ago by Nishimura's lab. And so what they did was they took a plant F-box protein, which is an auxin responsive component of ubiquitin ligase complex, and they
Uh-huh.
expressed it in yeast. And they found that if you could take another protein in yeast and fuse it to this auxin-induced Degron tag, then when you add auxin, it acts like the small molecule acts like a molecular glue to bring together the ubiquitin ligase complex with the tagged protein, and then that protein gets rapidly ubiquitinated and degraded by the proteasome.
Mhm.
So
When you
turns
say
out it works great
rapidly,
for most proteins. Sorry.
yeah. So when you say rapidly, like how fast is it going to happen?
So we did a time course
Uh-huh.
before we went ahead at the perfect timing. So I found out that the depletion can happen as early as 10 minutes for some transcription factors. Well, for others, it might take up to 10, 15, 20 minutes.
Uh-huh.
So, yeah, that's why we slide the window to see the optimal time point that fits well. Like it should deplete the protein and we should see immediate effects, but also prevents capturing the indirect effects.
Yeah, I think if you look at supplementary figure 1 in the paper, Linda Warfield, who also made like very important contributions to the paper, she did Western blots of 150 plus transcription
Wow.
factors and time courses, and you can see how fast each thing
Mm.
gets degraded.
Mm.
But anyway, we
Right.
picked 30 minutes because of gene expression changes and how long it took for the factor to get degraded, and we wanted everything to be reproducible, so we didn't want to just take right at the
Mhm.
point where it was getting degraded.
Mhm.
Just a little bit longer.
Right. And lastly, let's talk about 4TU pulse labeling. What is that? And again, how did you guys use that?
So, yeah, usually, before I joined this lab, I only know that people sequence RNA and see whether the gene is going up or down. But if there is a differences in the mRNA decay and its half-life, if there is an imbalance in there, it will also reflect in the changes. So that's not a direct effect on transcription, but the changes we are seeing is due to the changes in the mRNA decay. So that's why it's always more efficient to look at the nascent transcription rather than the steady state levels. So that's why Hahn Lab has been using 4TU labeling even before this project, and that eliminates the indirect effects that may arise due to the mRNA stability defects. And the way it works is that we deplete the transcription factor of interest for 30 minutes. And just before freezing the cells, we add 5-thiouracil, which is an analog for thymidine, and it gets incorporated into the cells, especially into the RNA that's being synthesized. And then we freeze the cells after 5 minutes, so it will just stop there. So we pull out only the nascent RNA, get rid of the old unlabeled RNA, and sequence this. So whatever changes we are seeing, that's in the nascent RNA. So that's definitely much better in a way that we are not capturing any indirect effects again.
Yeah, I think there's numerous examples in the literature now that if you measured the nascent RNA versus steady state RNA, you can definitely get different answers. So we want to make sure what we're interested in is the rate of transcription. So that's why we need to measure the newly synthesized RNA.
Right. I think like the common theme here is that most of these methods are fairly new, and this is why this paper was possible like now, and before this, like we didn't have all of these tools together to do like such a comprehensive study. US had some innovations when it comes to yeast construction as well, which I totally escaped. Can you like briefly talk about that as well? Maybe I can like so I guess like if I want to ask you a question, is you went for almost all transcription factors, right? So why did you miss some of them? Was there a technical challenge that you had to like skip some of them?
Yes, definitely there were technical challenges. I mean, we started with 212 transcription factors, and fusing MNAs to some of them resulted in slow growth. That's why we didn't pursue them. And for others, we didn't get a good quality check data. So that's why we dropped some of those. Yeah, those were the technical, and some of, I think we didn't get tag for some of the transcription factors, and we left them. Yeah.
Do you
Same
know
in-
off the top of your head, like how many TFs did you exclude from this study? Like, did you have to exclude?
Yeah, when you said 200, not all of them were actually transcription factors that have a DNA binding domain.
Yes, there are some cofactors and repressors too in that list, and some chromatin remodelers.
I would just off the top of my head, I'd say at least 90%. Yeah, and like the ones that didn't give good check data, like for example, yeast go into, just as one example, yeast go into this thing quiescence where they're just kind of in suspended animation almost. And some factors are required in that state, but we're not in that state, right? So they're probably not even expressed, and you wouldn't expect them to be binding DNA. So that things like that might be a reason why some of the factors didn't give good, binding data. So we grew the yeast, I like to say, in the most boring possible condition. They're in glucose, we're giving them all the stuff they need, right? So we can't deplete factors in every, 20 different conditions. That would take a long time. So
Yeah, great. So let's just start with the experiments, and let's start with figure 1. Can you walk us through the experiment that you did for figure 1, which is the genome-wide mapping of yeast TF binding?
Right. So the figure 1, it's mostly talking about the genome-wide mapping of yeast transcription factors. And we selected 178. Like Steve pointed out, not all of them are sequence specific. Some of them are repressors and chromatin remodelers and other coactivators, whereas most of them are categorized as sequence specific activators, and some of them are putative transcription factors. So when, like in yeast, we don't have enhancers. There are something called upstream activating sequences, which is a part of promoter. And it's known that transcription factors bind to these places, but we want to see the distribution from the transcription start site, like how are these binding site distributed from the TSS. So that's the B showing the distribution of these binding sites from TSS, and it's mostly in the promoter that we chose, that is 400 base pairs upstream and 200 base pair downstream of TSS. And like when we use this and used Homer for calling peaks, that is tenfold above MNAs, which is a control-free MNAs control, we saw thousands of binding sites for most of the transcription factors, which you can see in the figure C, the number of binding sites on x-axis and number of TFs on y-axis. So yeah, majority of them have thousands of binding sites. But the most interesting part from this figure is D, where we really want to see the presence or absence of motifs in the binding sites that we found for each transcription factor. And like you can see, most of it is yellow. That means it does not contain a motif, and very few of them that are in green contains motif, which is really surprising because that's not what I read about the sequence-specific transcription factor. I mean, it's for few factors like general transcription factors, I do see really good motif enrichment in the binding sites, but that's not the case with other transcription factors.
And this
And
is what
then
made you go with the other experiments which ultimately made this paper.
Yeah, I mean, we were surprised, but we also want to, actually, yeah, that's one of the reasons we want to see that it binds to thousands of these promoters, and many of them don't even have motifs, but we are interested to see if they act at these sites or it's just binding. And that's one thing we wanted to.
Yeah, right. Like, is its DNA binding domain important for going to sites with motif or without motif or both? And that's led Lakshmi into one of her other figures.
Yeah, just supplementary, but yeah, that's one of the figures too.
What about figure C? Were you like surprised to see that some targets have like 3,000 binding targets? Like,
yeah, we definitely were surprised to see that a transcription factor, sequence-specific transcription factor, which is supposed to act at only a couple of promoters, binds to half of the genome. But that could be because it might be sampling the genome to find its original target, and the check-seq is detecting those sampling targets too. That's one of the reasons that I always say, but yeah. And yeah, so
you also did, yeah, with E, you kind of compared the no motif and motif ones.
sorry, what's that again?
The figure E.
Yeah.
Yeah. So it's just I think it's very interesting because like you see D and you naturally ask, well, maybe the ones that are obeying the motif are just stronger. But yeah, you just showed in E that that's not really the case.
Yeah, that's what we expected, but that's not the case. As you can see in the E, that no matter whether it has motif or no motif, the check signal seems to be almost same, with few exceptions of one or two, but most of them obey this. But based on this binding data, we are interested to see if TFs prefer one class over the other class of promoters. That's why we did a k-means clustering, unsupervised clustering of gene classes, just based on these TFs binding, which actually revealed 3 interesting classes. Most of them are differed based on the number of TFs binding to its promoter. Like the class 1 barely binds with, like it has very few transcription factors bound to its promoter. Class 2, moderate number. But the most interesting class to me is cluster 3 because it binds numerous transcription factors to it. And these are actually enriched for those highly inducible promoters that Steve's just talking about. Whereas the cluster 1 and 2 are enriched for constitutive genes.
And these clusters are just defined by K and N. or
Yeah,
like K
just
means?
by k-means clustering based on our binding data.
Mhm. So as you mentioned in cluster 3, there seems to be an enrichment.
Mhm.
So if you just combine F and G together, it seems like most genes that TFs bind to are present near chromosome domains,
Mhm.
LCID boundaries.
Yeah.
I'm just wondering, because like this is the first time that I learned about this concept. What does that mean? Like the chromosome domain boundaries. What does that
So
mean? Yeah. And like, was that surprising to see?
yeah, that's surprising. I mentioned this because these transcription factors bound to, like many transcription factors bound to these cluster 3 genes. And I explained it in the later figures that most of these cluster 3 genes binding is mediated by non-DBD, at least in case of GCN4. And one explanation could be because if the two regions of chromosomes are far apart, but they are together because there is a looping.
Mhm.
And the point where the two separate chromatins bind together, that's called chromosomal interacting boundaries. And these TF cluster 3 genes are enriched at those boundaries. That's what I mean by those CIDs.
Yeah, so we were surprised. I can't tell you what it means, but it seems really interesting, whatever it is.
Yeah, we still don't know what it means, but we observe that these cluster 3 genes are enriched at those boundary elements.
Mhm. Okay, so that is the binding experiment. Let's also talk about the function experiment, which is figure 2.
Mhm.
Can you walk us through figure 2,
Yeah,
which
sure.
yeah.
I think I mentioned this to you when I was explaining about the auxin-induced Degron system, that we did
Yes.
time course to land on the optimal window to uh, get the direct targets at the same time, trying to avoid as much secondary effects as possible. So this figure explains the same. We took 4 different transcription factors and collected time points at 4 or 3 different time points.
Mm.
And at 30 minutes, we did see a number of genes going up and down. But when we collected samples at 60 and 120 minutes, some of the genes that were affected at the 30 minutes got recovered, and we saw additional targets, which could
Mhm.
imply that this could be the secondary effects, and cell is trying to compensate for the loss of the transcription factor. So that's why instead of choosing a greater time point for depletion, we landed on these 30 minutes. And that's exactly what it shows in the B. And so when we when we are trying to publish this paper, we got resistance from the reviewers saying that the effects that you are seeing at some genes where there is no binding, that it could be just an indirect effects causing by stress regulatory response or activation of some cell signaling. So that's why we want to remove those secondary effects. The way I did it is by looking at the number of regulatory TFs per gene,
Mhm.
which is plotted on x-axis in the C. And
Mhm.
I'm looking at the density of genes on the y-axis. You can see a tail coming off after 20 transcription factors. That means these genes are regulated by more than 20 transcription factors, which is deviating from a normal distribution. So this tail
Mhm.
could imply that this could be non-specific effects. I might be throwing out some important biological information here, but I want to minimize all those secondary effects. So that's why I did filtering, where I'm throwing out the genes that are regulated by greater than 21 transcription factors and only taking the ones which are below.
And that was based on the Z-score,
Yeah,
right?
and I did
Mhm.
this by a statistical method where I calculated z-score and threw away the genes that has a score greater than 2. Um, so then whatever remaining genes we had. I worked with those genes in the next figures. But the F
Yeah.
says something else. It's about, so as you can see in the heat maps, the genes are going up as well as down when I depleted the transcription factors. In this figure, you can see GCN4 and REB1 are traditionally categorized as activators, which means you should only see red but not blue, but you're seeing both in here. Likewise, CYC8 is a co-repressor, and but I'm seeing the genes going up and going down as well. We want to see if this holds true for all the transcription factors we assayed. So I put a cutoff, like if a transcription factor is resulting in the upregulation of genes, which is greater than 70%, then I categorize it as an activator. And if it is repressing greater than 70%, then it's a repressor. But anything in between, I categorize it as dual. So these
How
numbers
did
can be
you
seen.
how did you come up with the 70% number?
Oh, that's an arbitrary cutoff. I actually played around with more cutoffs too, but 70 seems to be more lenient. I don't want to go as high as 90 or 80% because none of them will be a pure activator or repressor if I take that high threshold. That's why I loosened it a bit. But yeah, that's no basis for that.
Yeah, there's no magical
No.
formula for 70%, but it seems like a reasonable number to us.
Yeah. I mean, like
But
looking at E, it seems like, any cutoff will just result in the same conclusion, really.
exactly, that's what I was about to say, that
Mm.
if you don't like 70, you can put any number here, but you can still see many transcription factors are in between where they are both activating and repressing genes here.
Right.
I'm not surprised to hear that a TF can act as both activator and repressor, but I'm surprised at the scale, like how many TFs can do this. This is really
Right. So
surprising.
this
for
is
us.
kind of like the first major surprising discovery that like most TFs have actually dual functions. They're not like binary activator or repressor.
Yeah.
I mean, we kind of talked about this, but again, like how does that compare to like the general perception of like TF function before like this paper?
Well, I think before, I mean, we definitely have known of examples of factors that could be activators or repressors for at least 30 years. But it was thought to be like a more of a special case, not the biggest class of how transcription factors work.
Right. And so when we say like the majority of TFs have dual roles, what does this actually look like in practice? Do you have any like specific example of how one factor might activate some genes and while repressing others?
Yeah. Well, in this work, we haven't shown how it works, but from the literature, we know, for example, in yeast, CBF1 is shown to have this dual function. At least one paper from Podstein lab, I think they said CBF inherently acts as a repressor by recruiting chromatin modifying factors. But when it's in complex with MET4 and MET28, that's when it starts acting as a repressor. So that's one model. They said that it could be, depending on that factor it interacts with, it could act as either activator or repressor. Sometimes, like for example, in higher eukaryotes like P53, people have shown it has something to do with the sequence, like based on the promoter context. Like if it binds to one sequence in a promoter, it acts as an activator, whereas at other promoter, it acts as repressor based on the sequence motif it binds to. So uh, we haven't shown it in this paper, but that's something we are interested to explore. But I think for now, I think it could be context specific depending on what TFs it interacts with, or it depends on a promoter class or the co-activator presence or even chromatin configuration.
All right, so moving on to figure 3. So now you have done the binding experiment and also the function experiment. How do you link these two to make the the main discovery of this paper, which is the title of the paper.
Yeah, well, this was really exciting part for me too. I got binding data and I got function data. All I
Sure.
want to do see is how much overlap is there and what kind of binding site is functional and all that. So this is what I'm showing in this figure. In the figure A, I'm showing the number of binding sites for each transcription factor that is shown on x-axis. On the y-axis, you can see the binding sites, but the one in the blue is it has a binding site, but also when the TF is depleted, the gene downstream to it is affected, either upregulated or downregulated. The overlap is good for few, in fact, very few TFs, but in the most cases, as you can see, a tiny bit of binding is only resulting in the function, whereas rest of the binding doesn't seem to have effect in its expression. And we looked at the other way around, like we saw the genes that are changing and how many of them do actually possess binding at its gene regulatory region. So that's what I'm showing in the B, total number of regulatory targets. And the blue are regulated and those ones have binding in its promoter region, whereas majority of them don't possess a binding, at least
That we can detect.
that we could detect from. Yeah. And we thought, oh, maybe we are growing cells and glucose rich, and some of these TFs might not act in these conditions. So that's why we might not be seeing a greater overlap. So we picked a handful of transcription factors and we grow them in conditions that they are known to act. Like, for example, GCN4 in SM, it's supposed to activate it. Likewise, some heat shock proteins at 37 and GAL4, which is well-characterized transcription factor, was grown under galactose-inducing conditions. But even under those conditions, we still saw greater number of non-functional binding sites, and we also found some regulatory targets for each TF, which do not possess a binding site for that corresponding transcription factor. So this is like you said, this is the main conclusion from our paper. Totally surprising that it holds true for most of the transcription factors.
The way that I understand this thing and think about it is that if we have a Venn diagram of binding and function, when there's low overlap, this means that we have so many bindings without function, and we have so many genes that are regulated without any binding. What does each mean, really? What does it each imply?
Okay. I don't have a great answer for you here, but I think the fact that there's a lot of binding that doesn't do anything, part of that probably has to do with the mechanism of how transcription factors find their DNA. For example, there's this one model called the Wolfpack model, I think we mentioned in the paper, where in that model, transcription factors can associate with each other via their disordered regions, and then they would go looking for binding sites as a group. So maybe binding is only mediated by one or a few number of transcription factors in that cluster. So maybe there's one or two important things that are maybe directly binding DNA, but the others are just kind of along for the ride and they don't necessarily do anything. So that would just be, for example, a consequence of how things bind. Another thing is, factors might be redundant with each other. So if you get rid of the factor, there's plenty of other factors to take the place of that function. Or maybe factor binds to DNA, but its biochemical function is not actually important for turning the gene on at that promoter. Like maybe you don't need to remodel chromatin at that particular promoter because it's already open to start with.
So, correct me if I'm wrong, but you're saying that there is still some underlying mechanism, even though we don't understand it. There is some mechanism, which means that the binding is not purely random.
Yeah, that's what I think. It's not random. if anything, it might at least reveal us how transcription factors might be sampling the genome. It might not be doing anything there, but it will tell us how TF might be trying to locate its actual target, like Steve mentioned about Wolfpack model, and they could. I mean, I might be just imagining it, but it could be sometimes the promoters might be acting as a reservoirs for TFs. Like it needs to be there such that when it's required, it can immediately go to the place of action. So they're just landing pads rather than always need to be there to activate the gene or request the gene.
I mean, it's a very different way than I learned how gene expression works. I mean, I think the assumption was a factor binds there, it's always going to regulate that gene. And like, we're definitely not saying that. And I think
Right.
other people are, when we start talking about this, other people that I talk to are like, oh, yeah, I saw that too, but I just didn't know what to make of it. Right. So you kind of, when you get data, you kind of naturally want to focus on the things you can understand and put the other stuff off to the side. And so I think by not just looking at one factor, but by looking at many factors and seeing that this is like a general behavior, it made us more confident of, presenting the results and made a more compelling case that this might be a general kind of mechanism for how transcription factors work.
You also did some fascinating experiments with GCN4, showing that its activation domain is important for DNA binding and not just activation. Can you explain how this challenges our understanding of how transcription factors find their targets?
Oh yeah. So like I said, I was seeing many binding sites without motif, and I really want to. I mean, at that point, some other lab started many labs are showing that non-DBDs also have a role in promoting binding to the promoter. So it's definitely they are not the first ones to report it, but that makes me wonder if that is true in my case. So that's why I took only one transcription factor this time and deleted activation domain, also mutated DBD. And so I was, frankly, even though I read all those papers, I was quite surprised to see many more binding sites, even in DBD mutant of GCN4. And I, and then I also deleted activation domain and saw the binding is basically reduced to like 80% of the binding sites were disappeared in that mutant. And if I look at the browser shot, it's a nice way, like wild type shows 10 peaks, activation domain shows 2 peaks, and DBD shows 8 peaks. It's a nice mirror image to see the AD and DBD mutants. So, and to my surprise, I saw that most of the peaks that I'm observing in DBD mutant lack GCN4 motif, whereas activation domain, a mutant which is basically getting there only via DBD has motif. So that's matching very well with the literature too. So yeah, I mean, actually, the funny thing is, it's just not intrinsically disordered region. When I mutated the key residues that are important for the activation of the GCN4, it still lost its binding at majority of promoters. That says that its interaction with the other factors might be important for the stability, I guess, DNA binding.
Or targeting one of, we can't
Yeah,
say at
yeah,
the
we cannot
moment.
say. I mean, at this point, we don't know that, but those are really interesting observations that got me excited.
Yeah, I think there's been, even though people talk about transcription factors use their DNA binding domain all the time, there's been a number of observations over the years that don't really fit with that. Just for example, like if you look at the size of the motif for most most eukaryotic factors, it's really small. And it doesn't seem like there's enough specificity just in the DNA binding domain to target it to some small number of genes, right? Um, there's a really interesting preprint from the TGen lab that just came out, and their theory is that as the eukaryotic genome is much larger than the prokaryotic genome. One strategy could have been to make the DNA binding domain much, much more specific, but instead they kind of took the opposite approach and used other parts of the protein, the disordered part, to help direct where it should go. So I think that theory
Interesting.
fits perfectly, I think, with what Lakshmi has found here and other people in the field too.
Let's not forget about the last figure, figure 4. So here, after the main result, you kind of like replaced the promoter region, and you moved it to a whole different chromosome, chromosome 1. Why did you do that, and what did you find?
Well, we did it because reviewers asked us to. But yeah,
Yeah, they didn't say do that, but they're like, oh, you're getting these things where you don't see transcription factors binding at the genes regulated. I need you to show me that's not some artifact.
yes.
Hmm.
So that's exactly how it started. But this is a really interesting approach and actually answered so many questions and gave us more questions to answer. So the way we did it is we took the promoter region, which I just mentioned, like 400 base pair upstream and 200 base pairs from TSS. And we took the genes that are being regulated by TS, but the ones where we did not see the binding. So we took the promoter region of those genes and cloned it upstream to a GFP reporter and cloned upstream to it and assayed for its expression. Interestingly, when we depleted the transcription factors, general transcription factors like ABF1 and CYC8, it seemed like promoter alone is sufficient. Although we did not see binding, that region alone is sufficient to drive or the to drive the regulation from that particular TF. Whereas in some sequence-specific transcription factors like STP1 and SUM1, we did not see the effects we observed in the native chromosome region. That could mean that it needs to be in its native location to be able to be regulated by those TFs. It could be from the long range interactions, like Steve mentioned before, or sometimes the genes needs to be closer, like the co-regulated genes need to be in a cluster to be able to act by those transcription factors. At this moment, we don't know that, but this assay gave us two classes of regulation. One, it could be acting through low occupancy binding sites, like in case of ABF1, or it could be from the long distance, and like in case of these sequence-specific TFs.
Mhm.
Yeah.
So this kind of shows that like the thing that you mentioned, long range regulation, like mechanism, something, um, which okay, so this goes against what people knew about yeast, which doesn't have any long range regulatory mechanism, right? And maybe I shouldn't be asking you this, but like, how did they come to that conclusion? Like, why they thought there's no long range mechanism in the first place?
Okay. So these upstream activation sequences were found actually by my postdoc advisor. So this was like 40 plus years ago. And upstream, okay, so enhancers are regulatory sequences that can function from variable distance upstream and downstream and relatively orientation independent. So the yeast UAS sequence, they're called upstream because they didn't function downstream of the gene. And when people move them around, at least the ones, people haven't looked at everyone up until recently, but they usually function within a few hundred base pairs from the gene. So, but of course, those were experiments where people are looking at things one by one rather than looking at everything kind of all at once. So we didn't want to use the term enhancer, which we don't use in the paper, because until it's actually proven, I don't want to be saying anything that could be wrong. But I mean, I think Lakshmi's data and Linda Warfield, who also really contributed to this too, I think their data says that there certainly is a possibility that the regulation isn't only coming from these localized sequences, but something far away that we don't understand yet.
Mhm. You also did another experiment, which I think we kind of skipped over, where you like introduced heat shock and or like amino acid starvation, and you showed that the general pattern still holds. Like there's a low overlap between binding and function. And I am curious to know why you did that. Was that um, like an attempt to make it easier to like apply the results from this like general like unicellular yeast to like more specialized human cells that kind of like have a direction in terms of gene expression, like you wanted to like introduce that direction into the system, or like
I mean, frankly, I saw a low overlap between the binding and regulation in most of the TFs. So one of the reason could be, like I mentioned, and I was explaining the figure 3, I said one of it could be because the TF might not be growing in a condition that it's supposed to act. Maybe that's the reason we are seeing a low overlap. So that's why I did those experiments in the starvation conditions and the heat shock conditions to see if the overlap improves if I grow the cells in the conditions that they're supposed to act. But that's, I mean, definitely there is a greater percentage of functional binding, but we still see a lot of non-functional binding and regulation without binding targets in those conditions too.
Right. If we go back to the binding clusters, so like we found that some genes or like some gene promoters are bound by over like 100 different transcription factors. So how do you envision this working like mechanistically in that like very tight space? Uh, like, do you think they all bind at the same time, or like is there like a I don't even know.
So these cluster 3 genes definitely have wide nucleosome depleted regions, but
But they're not
still
that
not that.
they're not that way.
Yeah, it's they have wide, but still not wide enough to be able to uh, recruit like more than 100 transcription factors, like you said. I mean, we don't have evidence, but we think there might be a dynamic exchange of transcription factors. They might not be binding all at a time, but few, maybe in some sets. Or we are assaying from bulk cells, like from all cell stages and different kind of, like we are not doing a single cell sequencing here. So we might be capturing information combined from all the cells, but maybe it's happening few at a time. We don't have that information.
Yeah, or things like, the wolf pack model where there's a big cluster of transcription factors, or there's another kind of similar model that people have that transcription factors are not equal, they're not homogeneously distributed throughout the nucleus, so they could be in clusters. So the gene could be recruited to one of those clusters. And because we're using MNase, that factor doesn't necessarily have to be directly bound to the DNA. It just has to be close by. So that could be another reason, like why we're seeing at these classes of genes many factors in the nearby vicinity.
Can you elaborate on this wolf pack model?
The wolf pack. So that was proposed by Max Stoller, who's at Berkeley. Um, and he came up with that as a way to try and explain how transcription factors find their binding sites, and the fact that people are finding that the disordered region of eukaryotic transcription factors are important. And that people, when you look by microscopy, not every factor, but some of them, they seem to be, distributed in clumps throughout the nucleus. So, like I said, the idea is that groups of factors will pre-associate, and then those groups will go around looking for binding sites. And like that's one thing that we're trying to follow up on in the lab, actually test that model. And I mean, if it's true, like, does a factor only associate with like one set of other factors, or could it be in multiple different clusters? And how does that behavior affect the activity, activation or repression or what have you? So, I mean, it's a really fascinating subject about how factors find their binding sites.
Right. So your data, um, maybe not like overtly support, but it doesn't challenge the Wolfpack model.
No, I'm not saying that's the model, right? But
Mhm.
it's that model is one of a few that are consistent
Mhm.
with the kind of data that Lakshmi's got.
Mhm. So well, as you said, I'm guessing like this is one of the follow-up steps that you want to go with this paper, just like the Wolfpack model and some other models you want to test.
Oh, sorry, I didn't hear your question.
So I'm guessing like the one of the follow-up steps of this paper would be testing all these different like Wolfpack models and things like that for transcription factor binding.
Yeah, so yeah, the way we're doing that, another postdoc in the lab, he's using a couple of different methods, one of which is proximity labeling. So if you have
Mhm.
um, biotin ligase associated with a certain transcription factor, and then you activate that, like how many other proteins in the growing cell are near that, how many are transcription factors, and when you figure out that set, like are are those factors, cooperating with each other either for DNA binding and or function,
Uh-huh.
right? So then there's other biochemical methods you can use to test it, but it's fairly straightforward actually. And then Lakshmi is trying to use microscopy to look at like how regulatory regions come together and if transcription factors are clustering, things like that.
Mhm. Um, so if you want to talk about the broader implications, your findings suggest that the traditional model of transcription factors working primarily through nearby binding sites needs serious revision. How do you think the field should be thinking about transcription factor function moving forward?
Uh, well, I think if anything, our paper says that just don't rely on motif and binding information to really know about its function. Always pair it with the regulation data as well.
Yeah, I think it's important to have both the combined binding and regulation data. I think there's many studies in the past that have just looked at binding data and tried to make conclusions about what things are regulating what genes. And I think, you're definitely missing important information if you do that.
Mhm. We said that lacks this like long range mechanism, a regulatory mechanism, which is found in mammals. So do you think if we do the same experiment in like human cells, would that like would the results would be even more pronounced?
Well, so in say human cells, for example, there are long range enhancers known. So if you see a transcription factor regulate some particular gene, but you don't see binding at the promoter, you know, the
Uh,
default answer is, oh, there's a long range
uh,
enhancer. But where is that? I mean, it's really difficult to find where that is coming from. So I
uh,
think that aspect, like, it's not surprising to anybody who works on humans that, oh, yeah, there's enhancers. I think the thing that is surprising, but I think people are starting to find it, is there's plenty of binding of transcription factors that doesn't regulate the gene, right? You'll
mhm.
see the binding and you take that factor away by some mechanism and nothing happens.
Hmm. Um, for someone developing drugs that target transcription factors, what are the practical implications of your work? Does it change how we should think about therapeutic targeting?
Uh, well, I would say when you're thinking about therapeutic targets, people already have an idea of what transcription factor they should target, either because there's a mutation in that factor that leads to the disease, or there's some signaling pathway that's messed up that you can directly link to a transcription factor. So I don't know that our work would change how you select your targets because it's based on actual information about a disease. But I think what it could do is like when people are trying to say, oh, there's this mutation and it affects gene expression, or it's in a particular part of a transcription factor that's not the DNA binding domain or the effector domain. I mean, I think our results are clearly relevant to that kind of thing, that it might help you understand why a mutation outside of the DNA binding or, say, activation domain leads to a defect, or why there's no motif or a particular transcription factor, yet that factor is acting at that gene.
And looking forward, what are the biggest questions your work raises that you're excited to tackle next?
Well, I would say, I'll let Lakshmi chime in here, but I think for me, the two biggest areas are that I'm interested in pursuing is, how do transcription factors find their targets on DNA? I mean, it used to seem such a simple question, and now it's like, well, it's really complicated. And then the other thing is, how do transcription factors combine their activity to generate a functional regulatory element? So, for example, there's very few transcription factors, like, say, P53, that are really strong and all by themselves can turn on transcription. But the usual thing people are finding that each transcription factor contributes only a little bit to the final output. And how does that work? And Lakshmi's got this giant table that says which factors work together or which factors work in opposition. And I think that's a great starting point to try and investigate how the factors, say, cooperate or help in repression, and investigate the mechanisms of how they do that. So those are the two general areas I'm interested in.
Yeah. In continuation of that, I'm also interested in that, but more specifically, I'm interested to see how transcription factors are targeted. I mean, especially at the cluster 3 genes, there are like hundreds of TFs. What's the basis, if not motif? Like, how are they going there? What's the scaffolding protein involved, if any? And then what's the biological significance? Like you asked, it's not transcription. What's the biological significance of that? That's the main focus of my future study. That's what I mean.
Amazing. Thank you so much for this paper and this conversation. It was lovely to have both of you. And if we can just end it with this question for both of you, what advice do you give me as a PhD student?
I would say, know the literature really well. Like, you can't put what your own work in context or figure out what important questions you want to address if you don't really know the literature really well. And the second, keep an open mind. Like, for example, when Lakshmi got this contrary data, I could have been like, Lakshmi, you don't know what you're doing, right? But, if you keep an open mind, and even though you know the literature, some fraction of it is not. Maybe it's specific for what that system is, but it's not really general. So you have to keep an open mind that some of what you read does not apply to anything. It's like a special case to however they did it. So you have to be open to new ideas. So those are my two pieces of advice.
And I agree with that.
What about you, Lakshmi? Do you have any things?
Yeah, I mean, other than literature and keep an open mind, I think, yeah, basically, I also have the same things actually. Nothing else to add.